Hi, my name is Jessica Redline, and I'm going to be showing you Lab 2, part of isolating the colonies. This is for LAPU Micro 225, and it is 7/18/2020. So in the first part of the exercise, I demonstrated the part where I got the cultures in the broth, and I have them in my bag here from the refrigerator from a couple days before, just preserving them. So we have E. coli, S. epidermidis, and the S. cerevisiae, the yeast one. I noticed that with my tubes, the S. epidermidis and the S. cerevisiae, E. coli and the S. epidermidis, they both developed like at the same rate. They maintained the same color of the broth, the natural broth color, and their turbidity was pretty low. It's still really clear and you can see through it. I noticed that the only cloudiness was towards the bottom of the tube, where you could see some growth happening. I think that the E. coli one was a little bit more dominant than the epidermidis one. You can still see like there was some floating particles, and also it had the cloudiness located at the bottom, but the top portion was clear still. And this is the S. cerevisiae, so this is developed into a completely different color. It's yellow, and it was originally the same color as these two, the light, like red tinged, pinkish broth color. So this one turned yellow, and this one seemed to develop a lot faster than these two samples. So I noticed that the turbidity in this one was pretty moderate. You can still kind of see that it's pretty cloudy. It just looks like a lot thicker, and it's a little bit clearer up here at the top of the tube, and then really cloudy towards the bottom. I also noticed that when I had incubated it for 48 hours, that at the bottom there was like a dome-like disc that was just sitting at the bottom, and it still had the cloudiness up at the top. So for the second part, when we developed the cultures into the agar plates, the samples came up with the streaking. So this is I'll start with the E. coli. So you can see the streaking on it, and you can also see like one big spot there of bacteria forming. And where I did the streaking, it's kind of like in a dotted pattern, but you can see where growth was occurring. The S. epidermidis shows a lot more streaking than the E. coli. You can see like all four quadrants being streaked. You can see like the lines of the streaking over on these three, and then it becomes speckled up at the top corners over here and more like spread out. I'm not sure if that's from transferring the cultures from each quadrant, and if that's why that occurred, but you can see the speckles and then where I streaked it. So this epidermidis one had a lot more growth than the E. coli. The next one is the S. cerevisiae one, the yeast. So this one had a lot of growth as well. It's really filled in right here on this portion, and then you can see where I streaked it here. And then there's also some spotted growth. It looks like really big specks up on this region, and then it's clear here, so I'm not sure why that happened. I might have not streaked it properly over here from this quadrant over, or vice versa. But there is still a lot of growth on this one, which makes sense because in the tubes there was a lot of growth showing faster than the other ones. I would say maybe E. coli and epidermidis took a little bit longer to develop, where the yeast, the S. cerevisiae one, was a lot faster. And that could be for, you know, a number of reasons. When we activated the yeast in the warm water and spread it out, and I read that the yeast likes to sit, you know, in the dark and at those temperature ranges, and it just grows rapidly from there. But other than that, these were the results of my TSA pores from the tubes and into the agar plates.